The production of this program is made possible thanks to the support of the following and viewers like you. Over the past century, life expectancy in North America has increased from an average of 47 ages to now more than 75 years. Along with this increase in years, cancer rates have also increased. According to Dr. Sam Aparicio of BC Cancer and the Don Rix Distinguished Keynote speaker at Genome BC's Decoding Cancer event, cancer is not exclusively a disease of aging, but cancer is much more prevalent in older people. Aparicio says age association happens because cancer is a disease of the genome. Namely, the instructions that are stored in our cells become corrupted over time and lead to deficiencies in the duplication instructions that then can lead to cancer. Genomics are key to understanding why cancer can manifest in our bodies, and genomics also play a vital role in decoding cancer. So in this mix, enter Noble Laureate Michael Smith, who in 1999 envisioned a genome center in British Columbia that could and would become a world leader in decoding and developing genomic-based therapies to address cancer. And it's worked. I've invited Dr. Sam Aparicio of BC Cancer to join me for a conversation that matters about addressing cancer through genomics and British Columbia's role as a global leader in research and treatment. We really are a global leader, aren't we?
We are, and we've been leading in genomics since Michael Smith established the Michael Smith BC Genome Sciences Center in 1999.
You know, at the end of your talk at the Don Rix Distinguished Speaker series, you said, remember, it's research. It really matters. Like being able to put this energy and effort into research that changes the future. When it comes to research and the role of Genome BC, how are we doing here? I know that we've established that we're a world leader, but how are we really leading the way?
You know, it does begin back with the foundation of the Genome Center at the time when Human Genome Project was midway. It was an epical moment because it really signaled that Canada and British Columbia wanted to be a leader in the field by recruiting research leaders. And my colleague Mark O'Mara and Steve Jones were recruited at that time to found the center. Over the last 19, 20 years plus, the GSC has, with the support of Genome BC, advanced not just the science of genomics in cancer, but in many other areas, and also helped develop technologies. And that's been crucial to establishing here a critical mass. We have a critical mass of researchers here in BC.
Let's talk about how complicated understanding the genome of cancer is, because from my perspective, every cancer must have its own genome.
That's right. One of the things we understand about cancer is it is a disease of mutations that happen in the genome. There are spelling mistakes or errors, if you will, that creep into the DNA every time a cell is dividing in our bodies, which is a normal part of life. Cells divide to keep us healthy all the time. Those errors, if they're not corrected, they can build up over time, and that's what gives rise to cancer. The complicated part is that over probably 100 years of genetics and genomics research, we've come to understand that there are over 3 billion letters in the 23 chromosomes that make up a human genome. So working out the order of those, even just for one human, was a Herculean task.
And now your genome is remarkably similar to mine, but it's not an exact copy. And how important is it, as you were as a researcher, to understand the specifics of your genome and its relationship to the cancer that you may have versus my genome and the cancer that I might have? And how do you do that?
Yeah, it turns out that the variation between individuals in their genomes is really important. And one of the very early things that was discovered in decoding the cancers of genomes is that we had to learn about the normal genome of each individual alongside the cancer, because every individual contains tens of thousands of differences in their genome. We're all 99.9% similar, but that 0.1% accounts for enough variation to be important, and to be important in understanding how a cancer might differ. So we end up having to decode two genomes every time we look at a patient's cancer, the normal genome and the cancer genome.
The production of this program is made possible thanks to the support of the following and viewers like you. And then how do you then determine, based on that information that you've been able to gather, what is the appropriate next step? Do you address the genome of the patient, or do you address the genome of the cancer, or do you do them simultaneously?
We do look at them simultaneously. For a proportion of patients, the normal genome of the patient, the germline is what it's referred to, that is important because, or can be important because for some individuals they carry cancer predisposing genes, so mutations that take away the normal function of a gene that protects the genome, and this gives rise to a higher risk of cancer. But for many patients, that isn't the direct comparison. What one is trying to do is understand what mutations has the cancer accumulated that are specific to the cancer, and that we get by subtracting the mutations from the germline from the cancer genome.
So are we talking about oncogenomics at this point in our discussion, or is that another element in all of this as we look at how we're moving forward and addressing cancer?
Yeah, this whole area is oncogenomics, and the way that we use genomic information, either from the germline or from sequencing a cancer, depends on a patient's situation. We've begun to learn about cancer-causing mutations that are acquired specifically in cancers that now are used to predict whether a particular therapy might actually affect the cancer. So in those situations, knowing about which mutations the cancer has will determine whether a treatment can be used. That's not the case for every cancer yet, but it's the case for quite a number. Yeah.
So let's say you identify in me a particular kind of cancer, and you go, okay, and you work with my genome to develop a therapeutic response. Does that mean that that response is going to be universal to everybody who has that kind of cancer, or does there have to be variations on a theme for each person?
Yeah, we know that for some therapies, having a mutation is a very good indication that a patient's likely to respond. There are mutations in receptors that determine how cells grow in our bodies, and pharmaceutical compounds, drugs have been developed that target specifically those mutations. In those cases, there's a very good idea that if you use that particular drug, whether the patient has a lung cancer or they have a bowel cancer, they're very likely to get a response. For other situations, it's less clear, and that's usually where we're somewhere at the boundary between what we actually understand about direct responses and what might be a response. It's kind of the gray zone in medicine.
When we take a look at the developments around the treatment of breast cancer that are a result of the research and work that's been done here, explain to me what that process of discovery was and what we've learned and how treatments are now being applied.
Yeah, the early part of discovery was actually finding patients who were willing to consent to have their tissues and medical records contributed to medical research. That's a huge part of what we do. We can't make advances, without patient participation. And then gathering materials, so tumor materials taken at the time of diagnosis or surgery, converting those materials into DNA we can analyze, and then sequencing the tumors. That's led us through a voyage of discovery with breast cancer, that we've discovered, breast cancer is not a single disease. It's a constellation of 10 or more different diseases where we can distinguish how a cancer might behave, whether it's more likely to metastasize, whether it might respond to a certain kind of therapy based on its genome.
And so where did that lead you with breast cancer and the development towards treatment that is having wonderful results?
Yeah, we've in the last few years begun to recognize in certain breast cancers, they're referred to as triple negative breast cancers because they lack expression of the estrogen receptor and they lack amplification of a gene called HER2, which is used for a different therapy in breast cancer. Those triple negative breast cancers are a very complex group of patients. And the genomics has told us that there are at least 2 or 3 subtypes of triple negative breast cancer where the genome itself is getting progressively more unstable. The DNA is beginning to mutate and fragment at a faster rate. And that's because these tumors are deficient in the DNA repair mechanisms that our normal cells have.
Within the patient?
Within the tumor.
Oh,
within
okay.
the tumor.
Oh, within the tumor. Okay.
Yeah, within the tumor. So the tumors acquire this deficiency by mutation, and it can be accelerated in patients that have germline mutations in DNA repair genes, such as the BRCA1 and BRCA2 gene. What we've discovered is that there are small molecule drugs that will bind to folded DNA. These are structures that form from not double helixes, but actually 4-stranded helixes, which is a DNA structure that was thought not to exist until I would say maybe 20 years ago. It was discovered that DNA could fold this way. And we've discovered that drugs that will bind to these structures can have activity specifically in breast cancers that have a deficiency in DNA repair. And we're trialing prototype drugs of that sort at the moment.
What kind of prototype drugs?
Well, these are drugs that we've developed that specifically bind to the folded DNA, and they cause breaks in a way that a cancer cell can't repair, but a normal cell can. And so we're exploiting that difference. And they're drugs that we think can be used in a population of patients that we identify with genomics.
So are these then drugs that you are in the process of developing, or are you working with existing drugs that have been approved?
These are completely new therapeutic entities. So at the moment, there are no licensed approved drugs of this particular class that have this action. It's a completely new concept in terms of targeting cancer. So the drugs we're working with, you could describe them as experimental or prototype drugs. They're drug-like molecules, but they're going through the stages of testing and evaluation to see how safe they are, how active they are, before they could be licensed as drugs.
The production of this program is made possible thanks to the support of the following and viewers like you. Here's Dr. Michael Smith going, okay, let's have this genomic center. Let's use genomics in its application to understand cancer and how we can treat it. But then one of the benefits of that, and probably not an intended consequence at the time, but now we're seeing happen, is that you're developing a drug that could then also be developed here in British Columbia. And it fits into this ecosystem of excellence and in the culture of life sciences developments in British Columbia.
I think it's an interesting observation, and at the time that the Genome Center was set up, it would be fair to say that cancer was a rationale for having genomics, but cancer wasn't a tractable problem. So it's almost like somebody was building a bridge, but we didn't know if we could build the bridge all the way to the other side. At the time that was started, but with vision and with energy, anything is possible.
I'm also, of course, drawn to the fact that it was Dr. Alan Eves who, when he was at the Terry Fox Research Lab, was starting to develop the medium in which so many of these different experiments could happen, which has led to the development of stem cell technologies. And how important is that in the work that's happening here in British Columbia?
Yeah, it's another great development that's come out of the BC Cancer environment, and we all depend on those kinds of technologies and reagents. It's a world force in biomedicine, and has certainly helped accelerate the research of many labs.
So let's come back to sort of the Genomics Center here, and we've talked about breast cancer, but where else is research underway right at the moment that is going to make, continue to put a focus on what we're doing here and the influence that we can have globally in helping with better health outcomes, hopefully for millions of people?
Yes, our group, we've focused a lot on breast cancer and to a certain extent on ovarian cancers. We're beginning to look at many other classes of cancer where we understand that the particular way that mutations accumulate in the genome may be important. These include bone cancers like osteosarcomas. More broadly in BC, there's important research happening in prostate cancer, in lung cancer, in many of the cancer areas, all of which are utilizing the critical mass and the expertise of genomics. And the genomics is orientating us in many different ways. We're learning about how to monitor and detect cancer. We're learning how to treat cancer. We're learning about ways in which we might think about risk, individual risk, as well as the way that our environments might contribute to the risk of cancer development, and where cancer originates from, the very earliest changes that we can see in our bodies. So in a sense, we're studying aging processes that are not yet cancer, but may lead to cancer.
You know, I remember many years ago being at a talk that I believe Dr. Michael Hayden had given shortly after the mapping of the human genome, and he envisioned a day in which we would be able to take a look at somebody's genome and go, okay, I see a pending problem. Let's intervene now to prevent that from happening. One of the things that I also recognize that since those sort of halcyon days, or heady days, let's say, we've come to realize, okay, mapping the genome is only one part of it. How do we move in the direction to have this kind of treatment? Is this a concept that's still possible, that we can anticipate where there might be a breakdown within a certain person's duplication code? We can say, we can intervene now and prevent that breakdown from happening.
We certainly know about mutations, variations that increase risk, and those are in use right now. They're in use actually in breast and ovarian cancer and a number of cancers where we know from genetic association studies that individuals that carry a particular gene allele, although they may be healthy, they are at a much elevated risk of developing certain cancers. And the impact of that is that these individuals can be screened more frequently, they can be monitored more frequently. There are certain interventions at a certain stage of life, like risk-reducing surgery to remove epithelium that might be at risk of developing a cancer, which will drop someone's risk of developing a life-threatening cancer later on in their lives. So some of those principles are already working. Where I'd say we're still on the edge of research is learning about common variation risk. So the kind of risk I'm describing, maybe that's 5 or 10% of the population, but we know that there's an inherited susceptibility, or can be an inherited susceptibility to certain cancer types, but it comes from many, many genes in the genome. And we're still trying to work out how many genes that is and how do we actually interpret the variation. That's really a kind of a leading edge piece of research that needs to happen.
The production of this program is made possible thanks to the support of the following and viewers like you. One of the other components, and I'm hoping that you can shed light on this for me because I'm a layperson around this, is I hear that yes, you may have, you're born with a particular genomic code in your body, but that epigenetics plays a role in how your own genome is expressed. Is that true?
Yes, broadly speaking, that's a good analogy. The epigenome has to do with the way that our genes are wrapped up and packaged inside cells. And in a way, you can think about the epigenome as determining when and where genes might get turned on and off as cells function. The code for that is, in a sense, actually embedded in the genome. It's very complicated to read it. And we're just beginning to develop techniques now that allow us to read these epigenomic marks, the associated factors that will tell us whether in the future a gene is going to switch on and off in response. And this is turning out to be very important in understanding what makes one cell a cell, how cells become resistant to drugs, also probably very much involves the epigenome.
So has genomics brought us to a point where we know, like with lung cancer, you're going to reduce the likelihood of lung cancer if you don't smoke.
Yeah.
Like, I mean, that would be a direct lifestyle change.
Right.
But has genomics helped us to understand, well, if you do these things, it is going to reduce the likelihood that you could have pancreatic cancer or that you would have bowel cancer? And because it's so complicated, are there like messages or lessons that we're already learning as a result of genomics that we can apply to our own lifestyle that could hopefully help us in our journey as we age? Because as we talked about, cancer being actually a disease of the genome. What can we do to aid our own genome?
To aid our genome. So we know about a number of environmental factors that interact with the genome. You've mentioned one very important one, and that's smoking. In North America, the rates of cancer have been falling, but particularly for lung cancer, and everyone associates lung cancer with smoking, but not just lung cancer. In fact, the risk of all cancers is increased by smoking. So we know about that factor. We also are now beginning to appreciate that a healthy body weight is a factor. The way that our bodies metabolize sugar has to do with insulin, and insulin acts on all cells of our body. It's also a hormone that can be a mitogen. It causes cells to divide. And we know that too much insulin signaling chronically throughout life can create an increased risk of cancer. That's one of the other effects. Alcohol is another one. We all like a glass of wine, but too much alcohol is another factor that has been associated with long-term damage effects on the genome, and which can predispose to cancer. So there are quite a number of these. Other risk factors that we know about, some of them are now actually very preventable. Viral causes of cancer, for example, for cervical cancer. There are now great vaccines that in the young adult population will block the transmission of HPV, which is the virus which causes most of those types of cancer.
A healthy lifestyle.
A healthy lifestyle, yeah.
A good diet.
A good diet, and take advantage of screening, and think about family history. Yeah.
Does frame of mind play a role?
Oh, that's a hard one to answer.
Yes.
I think it's certainly important to have a balanced and healthy perspective about the risk of cancer. I think hard to say whether it will really influence whether someone gets cancer, but certainly one's approach to health, and if unfortunate enough to be illness, then certainly a state of mind is important.
Well, thank you for giving us some insights into where we're at, and especially the very important role that I think genomics is playing in helping us to understand and move forward in treatment of cancer, because it is something that millions and millions of people are faced with. Thank you.
Thank you.